There are places in the world where 15% of electrical energy goes into data centers. If all the data centers were a country in the world, it would be the 5th largest consumer of electricity in the world. So all of humanity's data, which fits into 8,000 huge data center warehouses, plus everything in small companies, medium companies, and so on, including data held by private individuals, all of that fits in a van. The conventional method in data centers hit a very hard wall in 2040.
Modifying living organisms like we modify software, creating bacteria that produce medicine, or even algae that produce certain materials, synthetic biology is extremely interesting and opens up many possibilities. To be able to answer all our questions and to go deeper into this subject, François Képes is our guest today. Hello, François.
Hello.
So we have a lot of questions to ask you about the field. Can we first go back to what synthetic biology is?
Yes, of course. So, synthetic biology is a field at the intersection of biology and other disciplines like mathematics and computer science, and it has developed over the past 20 years, actually, based on concepts of the maturation of biotechnology, meaning technology based on living organisms, in the sense that technologies like those in chemistry and computer science are mature and allow for a number of things. We can come back to the keywords of what a mature technology is later, but without going into details yet, biology was far from it. Synthetic biology set out 20 years ago to approach this capability, the level of maturity other technologies have reached. And I think we can say with a bit of hindsight that it has come close without yet reaching the goal.
Okay. To give a bit of context, why do we associate this concept of biology with the concept of technology? What bridge is there between the two?
Well, indeed, biology and our understanding of it have allowed for more, how should I say, more applied, more active advances, in which we use the properties of living things or to use elements coming from living things to establish new approaches, create new medicines, produce materials at a lower cost and also at a lower environmental cost, and so on. So that's the general idea. Biotechnology is absolutely nothing new, since Noah fermented grape juice into wine more than 5,000 years ago, after the flood. It simply improved little by little. There was the trial of the pastors for winemaking, and so on. And now I would still say that biotechnology has gained a great ability to accelerate the production of chemical compounds or processes that have high added value. And it has gone far beyond the first steps.
All right, so actually we've always more or less used biology to help us. I mean, through fermentation, as we just mentioned earlier. Actually, what we're doing here is just one more step, an extra step in the way we interact, in the way we actually evolve with these. Yes.
These beings.
So, how can we actually use biotechnologies today to address different problems? What are the use cases that can be addressed by synthetic biology?
It seems to me that it responds to society's needs in one way or another, because if in the end there isn't a client or a user, then there is no need for the technology. Now, I think many of us want more personalized medicine, gentler approaches, and chemistry that is more environmentally conscious. And also maybe solutions to deal with climate change, to droughts or pathogens, to the pathogens that attack crops, and so on. So, it's in response to this concern that many of us share that synthetic biology positions itself as a potential solution for different fields. And maybe we'll also be able to talk later about the reason why it can address several fields and not just one field. And by the way, I think we wouldn't talk about it as much if it were a solution limited to a very narrow use case. It's actually something that has a breadth comparable to nanosciences and nanotechnologies. Since there is both science and technology in synthetic biology, and since nanosciences and nanotechnologies offer new potentials but without, how should I say, being compostable, there you go. To put it simply, synthetic biology offers others, but they are, in quotes, compostable.
What do we mean by compostable?
What we mean is that I don't know if you, as a computer scientist, have ever tried to put your tablet in the ground to see how it would break down. It takes a long time for it to decompose. If you put your yeast, which was used to produce a medicine, you kill them and put them in the ground, well, it's just going to enrich the soil a little bit, but it's not going to do anything else. It's not going to, they won't last long. They will be recycled immediately. So that's why forecasts are often quite optimistic, saying that synthetic biology in the end will represent a large part of everything humans manufacture, and probably greater in the long run in volume and value than what nanotechnology and nanoscience represent, which have that limitation, that it is not easily recyclable.
Okay, so what are some concrete applications that biotechnology is currently working on, for example?
Listen, we can take examples from several fields. One field could be medicine. There is a drug. I'm sorry, I'm checking because I have a hard time remembering the names. There's the compound called Januvia, and its real name is it's sitagliptin, which is produced by Merck and helps increase insulin production. Insulin in diabetics, so it's an anti-diabetic drug that now exceeds 10 million prescriptions per year, you see, and so it's recent but already a form of success. Another example from the medical field seems interesting to mention because basically we can clearly see in the medical world that we are moving more and more toward personalized medicine, yes, and there is reason to believe that it will eventually become personalized down to the individual.
Okay.
And actually, there is already an example that goes in this direction. You know that there are white blood cells circulating in our blood. Some of them are immune system cells called T cells. And these T cells, in fact, are able to attach to a target and damage it. In some cases of cancer, which we are not able to treat easily, it would be enough for these T cells to know how to recognize the right target, that is the cancer cell, in order to get rid of it. So we use these T cells, the CAR T cells will actually be taken from the patient. That is to say, in fact, we simply take a blood sample and immediately re-inject the blood, but without the large cells, meaning the white blood cells. And we treat the T cells so that they express a receptor on the surface of the white blood cell, which will be able to recognize the right target, typically the tumor cell, and we re-inject these cells into the same patient. So if this isn't personalized medicine, then what is personalized medicine? You see, it comes out of the patient, it goes back into the patient, and we think that maybe eventually we'll be able to do it almost in real time. That means, in fact, the patient could stay so we can give him back his cells. He wouldn't need to go home and come back a few days later. So it's a highly personalized medicine that's used to treat, well, that's pretty natural. Most often, for now, blood cancers, but also some solid tumors and also some autoimmune diseases like sclerosis. So here's an example that shows us how, alongside pharmaceutical blockbusters, as we say, like paracetamol, which isn't very personalized, there are treatments now that concern small groups of patients, or even treatments that are almost at the individual level. And there are good reasons to think, if we look at what has happened recently, that we are moving more and more in that direction.
All right. So basically, when we're talking about these treatments, we're talking about programming cells, about reprogramming cells.
Yes.
To help guide them either towards certain cells or to accomplish specific tasks.
Exactly. And this, but through limited genetic reprogramming, by the way. In this specific case of CAR-T, it's simply about making them express a modified surface receptor so they can recognize the target, so it's quite limited. Now, as I was telling you earlier, the applications in synthetic biology are quite broad. I can maybe give a recent example as well concerning food. Well, we have this whole issue with people who are vegetarians and so on, but who nevertheless enjoy eating good things, and that's their right. And so there's an American company, I believe, called Impossible Foods, that makes vegetarian burgers. Well, up to this point, nothing very special. But this burger bleeds. So let's be clear, because this is about synthetic biology. Actually, in blood, it's hemoglobin that gives it the red color, as you know. Here, soy is able to synthesize what's called leg hemoglobin, a plant-based hemoglobin. We call it leg hemoglobin. And actually, it's not, let's say, commercially viable, but it was possible to transfer the gene that codes for leg hemoglobin into yeast. And this yeast, we can produce enough of it and at a low enough cost so that we can introduce it into a food product. And so we end up with the burgers in question. So that's an example taken from the food industry. Another example that we sometimes discuss at the French Academy of Agriculture, because it's a really important point, is the issue of nitrogen fertilizers. You know that there has been the Green Revolution since the 1960s, which made it possible that today, even though there are more humans on Earth, we have much less famine. Even though there is still too much of it. And part of this success, a large part of this success, is due to fertilizers, especially the addition of nitrogen to the soil. These nitrogen fertilizers are mineral fertilizers that require extraction, transportation, and so their impact. And then they also have to be spread in the field. So the carbon footprint is really not very good, and solutions have been sought. So there's an interesting solution called Proven, which means to prove in English. Proven then is offered by Pivot Bio. So it's a liquid fertilizer that actually involves spreading bacteria that are used to associating with plant roots. In this case, there are cereals, and there's nothing really new about that. But the problem is that while they are able to fix atmospheric nitrogen and thus avoid the use of mineral nitrogen fertilizers, they don't do it when they are associated with the roots of this cereal. And here synthetic biology specifically intervened to modify this natural bacterium so that it can, or at least its genes are activated even when it is at the root of the cereal. And that's all, but it makes all the difference, because as a result, it will capture atmospheric nitrogen. It will make it usable by the root. And we no longer need mineral fertilizers, which also had other drawbacks, like the fact that excess nitrogen fertilizers would seep into the soil with the rain and so on, into the ponds. In the ponds, they cause the proliferation of algae, sometimes even toxic ones. In aquifers, they are sometimes present in excess. So here, this phenomenon no longer occurs with this solution. So it's interesting to see how we can respond. Biosynthesis allows us to address different issues.
It's true that it's extremely interesting, because here we've talked about several levels of application in health, in medicine, and even in the environment with bacteria attached to the roots. And it's interesting because sometimes when we say synthetic biology, it tends to worry some people because it triggers a whole set of ideas behind it. Um, what are the most common misconceptions and what can we say about them?
Yes, you're right. There were actually a lot of debates in the early years, to the point that it's the only scientific field I know where there were more articles on sociology or analysis than scientific articles directly related to synthetic biology. That's less true now. There are fewer questions, though some remain, which is actually normal, because with every application, you can always ask about its value, its benefits, its risks, and the balance between benefit and harm. That's the least one can do, so why not? So this is where the fact that the French Anglicism biologie synthétique isn't a perfect term comes in. Not just because it's an Anglicism actually, but because it brings us more to the idea of artificiality. Whereas in fact, synthesis can mean either artificial like synthetic or manufacturing. That is, it goes through a process of development, of manufacturing, which turns out to be a much better description for synthetic biology, in which there is a desire to manufacture things in a rational way, possibly even from a hierarchical perspective, where you build a device and then you put several devices together to make a machine. Yes, or in this case, it could be a bacterium that attaches itself to the roots of the cereal plant. In short, it turns out that this is really the angle from which synthetic biology is best described. Whereas if we say artificial biology, meaning synthetic, artificial biology, we say, well, if I sum it up, it's bioartificial. Whereas for me, bio means what I find at a certain grocer, and that's it. So there's a kind of inconsistency there, and that also raises questions on that front. So in the end, what can we really say? At the level of fundamental research, it's not necessarily different from what we used to do before, like molecular biology and so on. Mathematical models, computer simulations that allow us to design the product. Then, so I should have said that afterwards. The construction itself often involves molecular biology or synthetic chemistry, and then afterwards we move on to characterization using the same tools, chemistry or molecular biology. And that's actually what forms what we call the 3 C's, which are conception, construction, and characterization. It forms the loop of any self-respecting engineering process, except that in synthetic biology, we've added a 4th element, learning. And why? Because precisely we still don't have a fully mature engineering discipline. So actually, when we've gone through the loop of the 3 C's, we end up with a characterized result that more or less deviates from what we originally wanted. And we have to go through it again, maybe even 2 more times. And that's where the 4th element comes in, learning, which tells us, hey, I'm closer to the goal, but I'm not there yet. So I revise my mathematical parameters and go back into a loop of design, construction and characterization, but with parameters that are closer to the target. And so I finally get closer to what I want. And so the fact that there is learning is itself proof that we're dealing with a mature technology like computing, electronics, chemistry, and so on.
To take a quick look under the hood, here we're still talking about things that are on a very, very small scale. We're also talking about genetic codes. How can we work at these scales and be able to modify a genetic code that's both so long and at the same time so complex? Because if we could unfold it, we could actually stretch it out over several kilometers.
Yes, that's true. That's right. Yes. So you see, maybe here, we will have to differentiate between microbes, microorganisms like yeast or bacteria, for which the tools have existed since the 1970s. These are what we call molecular biology or genetic engineering tools, and they're still extremely valuable. But if you now look more at the larger cells, like our cells or those of animals and plants, these large cells have much bigger and much more complex chromosomes. It's that. And in that case, the traditional tools, let's say, are not enough. In general, if we want to act precisely, for example, on a targeted cell whose code we want to modify for its surface receptor. So some recent tools have allowed us to make better progress. We can mention the talons, but I won't go into that because the main thing, the key word today, is still CRISPR. CRISPR can be used on microbes, but it's not necessarily required, far from it actually. But on large cells, it makes a huge difference. So, as you probably know, the article that started it all was published in 2012 by the French woman Emmanuelle Charpentier, who wasn't working in France, and Jennifer Doudna in the United States. And they received the Nobel Prize in Chemistry for this reason in 2020. Okay, bon. Alors, in the meantime, a lot of things happened, and there was a huge surge of excitement. Because people, since 2012, have seen the incredible potential of this approach, especially for making precise modifications to complex genomes, like those of our cells, or let's say, the cells of animals or plants. First, just a quick word. So CRISPR is a system that allows you to target a protein called Cas9 to the right place, the desired location, on a genome, on a chromosome. So, in my first example, we're going to target the Cas9 protein to the region that regulates the expression of a gene, meaning how much protein will be made from that gene. Except that we've modified Cas9 so that it has an effect on regulation. In fact, originally Cas9 cleaves, it cuts the DNA. Here, it doesn't cut it, but it has a regulatory effect. And so we'll be able to activate or deactivate a gene at will, and whichever gene we want, since we target it using the CRISPR system, using a modified Cas9 protein. So that's a case where we're working on regulating a gene. Another case involves Cas9, which cuts and also allows for other possibilities. Then there are repairs that take place, and we end up with DNA that is no longer cut after using the CRISPR-Cas9 system. This is a case where we can introduce a modification, a mutation at will with a lot of precision. And here I might give a surprising example because this has been done in many contexts. And especially for cells that we make work, that we grow in the laboratory. But there's a slightly different case that brings us back to the medical field. So let me explain in a few words, if I'm allowed.
Yes, go ahead.
With great pleasure. Well, so here it is. It's about the issue of organ transplants. You are surely aware that there are far more people waiting for organ transplants than there are usable organ donations. I don't know, for kidneys, for example, there are 4 to 5 times more requests than there are available organs. Yes, for example. So the waiting times for transplants are very long, and sometimes we can't prevent a patient's death and so on. It's actually quite dramatic. So we've known for a long time that pigs have organs that are quite compatible in size and shape with humans. And even so, things have remained at a standstill. Well, there have been a few trials on monkeys, but it stayed at a standstill for 2 reasons. One reason is that there are retroviruses integrated into the pig's genome, and they have no effect. They're remnants, fossils from another era, from ancient viruses, but they can reactivate in humans. That means if we transplant a pig organ into a human, the virus can reactivate and cause damage. So that's one point. The other concerns the structural aspect of the surface proteins, which I won't discuss, but it's been well addressed since then. In any case, in 2015, in a single article, Church's team in the United States removed 62 retroviruses from the pig genome, all those that had been identified. I'm saying in a single experiment, and that's not possible without synthetic biology, and the tools that have been developed since it was created in 2004, it's unthinkable. In 2006, 2010, maybe even 2008, no one would have said it's possible or that it would ever be possible. And so suddenly half of the problem of organ transplants from pigs was solved. The other part has been partly solved since then. You know, you may have read in the newspapers like I did that there were 2 pig heart transplants in terminally ill cardiac patients, and they didn't work for very long. But if we look back at the first human heart transplants, which happened decades ago, the first ones didn't work very well either. And since then, it has become, I wouldn't say routine, but at least a procedure that is attempted in many hospitals around the world. So we can see how this radically changes perspectives and possibilities. But also, it seems to me, the characteristic time needed to get a result with these new approaches made possible by synthetic biology.
Earlier, you also told us about organisms capable of producing materials.
Yes, materials indeed. So there's an example, it's spider silk, which has exceptional mechanical properties. And which we've been able to transfer into different organisms, where it's actually much easier to control and harvest. So without specifics, since I don't know them, there are lower fungi that can synthesize a kind of glue and can actually be used to build a wall out of bricks. Well, out of whatever you want, by forming the glue that's needed for that.
So, another point that you've also worked on is information storage, either with polymers or DNA. Can you explain to us what that involves?
You know, a lot of people know that actually digital data is abundant and that it doubles in quantity every 23 years. That already, there are as many 0 and 1 bits in human digital data as there are grains of sand on the planet. Well, we haven't counted exactly, but that's the rough estimate. Or the number of planets in the known universe too. That's about the number. And so the issue is when we look at how this data is stored, just to assume that we keep having this doubling of digital data, big data, every 3 years actually, where are we headed? Today we're told it's generally stored in data centers, which are essentially huge warehouse-like facilities that house thousands of servers, basically computers without screens, and of course hard drives, magnetic tapes, SSDs, anything you want for storage. And of course we can only admire the underlying technology, as well as the fact that it continues to improve quite rapidly, even now. But we also have to acknowledge that there are places in the world where 15% of the electricity goes into data centers, where today's projects, and that's what we have nowadays, are being developed with a nuclear power plant next door, next to its data center, or maybe both together in order to power it. Aujourd'hui, if all the data centers in the world were a country, it would be the 5th largest consumer of electricity in the world, somewhere between Japan and India, and once again with a constant and rapid increase. So, at the French Academy of Technologies, we asked ourselves if there were any alternatives. This led me to lead a working group from the end of 2018 until the end of 2020, when we submitted our report, which was addressing exactly this question. So there are nanolithography approaches, which are interesting but not very easy to scale up. and we ended up focusing on a solution that will probably scale up and that already has some initial implementations. And in fact, it's about writing digital data onto DNA, the same chemical structure as our chromosomes. Even though we quickly realized that there were alternatives. So maybe just a quick word about the principle so that it's also clear why we have alternatives. Actually, the idea is very simple. When you create what is called a polymer, it can be a plastic material, it can be polyethylene, so CH2, 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 CH2. You can see that you won't be able to code ones and zeros since it's always CH2. But as soon as you use two building blocks called monomers to make your linear strand, which is called the polymer, you can say that block A will represent 0 and block B will represent 1. Absolutely. So then, what are the conditions for success? Well, you need to be able to grow your polymer, to add your monomers in the order you decide, meaning the order dictated by the sequence of 0s and 1s in your computer file that you want to encode, okay? Because if it's random, it won't work. So, there are technologies now that exist to meet exactly this challenge and in which we will be able to add as we wish. Along the unbranched strand with no branching, just a linear chain, we're going to put A's and B's, which represent zeros and ones, according to what the computer file we're encoding says. Yeah, okay. So DNA is a polymer made of 4 building blocks. So it has a 4-letter alphabet, A, G, C, and T, which means that in fact, we can potentially encode 2 bits per letter, right? Since, for example, that's the coding that was used. It's not necessarily the one that will become the standard, but we can say that A is 0, 0, C is 0, 1, G is 1, 0, and T is 1, 1. And from there, you make all your digital files, you encode them. So this question of coding is one that hasn't been completely solved yet because we can think of different improvements regarding this and regarding the objectives. But in any case, it exists, it can be used. But behind the coding, which is computer science, you obviously have to do chemistry. You'll have to synthesize the DNA. And that we've known how to do since the 1980s, except that it's too slow and too expensive, even today. But prices are plummeting. We're gaining a factor of 2 almost every 6 months with DNA technologies, which compares to gains of a factor of 2 about every 2 years. In computing, whether it's for storage, processing, or miniaturization. The numbers I'm giving are approximate, but it gives you an idea that indeed there is a possible path here, since the drawbacks, meaning the cost and slowness of DNA synthesis, will gradually become less significant. An approximate calculation shows that as early as this year, niche markets are possible. And that around 2035, 2038 in principle, we will be able to expand, let's say, to applications that today use magnetic tapes. That is to say, relatively slow and relatively slow, large-scale storage. Well, as I said, we do computer encoding, then we synthesize DNA chemically and preferably biochemically. It's less costly for the environment. Then we store it, I'll come back to that, then we read it. And so we've known how to do that since the 1970s. But we've definitely improved the methods. And then finally we decode, which means that where we have an A, we'll say it's 0, for example. Just the inverse operation that is done on the computer. So here are the 5 necessary operations. In terms of information density, we gain a factor of about 10 million. That's huge.
When we say informational, do we mean the ability to store information on the surface?
So in terms of volume, relative to volume, yes, compared to the volume. So all of humanity's data, which fits into 8,000 centers, 8,000 huge data center warehouses, plus everything in small companies, medium companies, and so on, and in private homes. All of that fits in a van, if it's on DNA. So it's not just a gain in information density, but it's probably one of the most impressive things in terms of power consumption. Different estimates have been made, all of which conclude that there's a gain of a factor of 1,000 or more. Next, there's the question of robustness and durability. You probably know that in a data center, data is changed and copied every 3 to 7 years, let's say every 5 years, to keep it simple. So magnetic tapes and hard drives are tested regularly, and when the number of errors gets too high and the repair system is likely to start failing, we copy the data over. So every 5 years. So here with DNA, we don't have that issue. In the lab, after 50,000 years at 25 degrees Celsius, we still have half of the DNA in good condition. Okay, so that means several things. First, it means that at 25 degrees, there's no need for refrigeration. So we don't use any electricity. We're not dependent on whether a generator breaks down one day. Refrigerators are replaced every 10 years in laboratories. So even that is a problem. And then it's expensive. This system doesn't consume any energy at all. Of course, there is some, but it's when you synthesize it, and when you read it, when you write it, and when you read it. But there isn't any during storage. And so that's 50,000 years. It might seem surprising, but if you think about it, we've unearthed mammoth corpses in the Siberian permafrost. And we've been able to sequence a good part of their DNA, compare it to that of the modern elephant, and so on and so on. Horses too, the ancestors of horses, the oldest one was about 700,000 to 800,000 years old. We sequenced it and so on. So yes, there is an extraordinary longevity, but there's also another aspect, which is durability, meaning basically the standards and so on. I remember still the 5-inch floppies. Then there were the small ones, the 3.5-inch discs, I think, and so on. Well, now we use SSDs in
hard drives.
Yes, it can be safe. It replaces hard drives, etc. That's it. So we went through a lot of steps. And if today I ask you if you kept your 5-inch drive, you'll probably say, not really. We have to make room. So you see that there's not only obsolescence, but there's also a loss, ultimately, a loss of equipment, of know-how, and so on. Well, with DNA, we have a big advantage. It's that as long as there is modern medicine, since our hereditary material is made of DNA, we'll know how to read and write it. So there's no obsolescence to anticipate. It's this whole set of advantages that we've highlighted, as well as the residual problems that exist, which are numerous, that we've emphasized in our report. And from there was already work being done in several countries, and work that started in France as well, with some initial successes, both in the private sector, where there are small companies trying to make progress in the field, and in the public sector, where researchers are trying to improve certain things a bit more fundamentally. On the public research side, there's a small exploratory program with a €20 million budget that began 2 years ago and will continue for another 5 years until 2030. And there is a company, several companies, for example, Biomemory in Paris, which is connected to Sorbonne University, but which is a startup, as we say, that is actively working in this field and has already had some initial successes. That's it.
Okay, so here we're really talking about something that has real-world applications today.
So today, with the first commercial solutions, again, limited to niche markets, heritage, anything that doesn't need to be changed all the time, cultural heritage, The institution that is, how should I say, the recipient of the work from the French exploratory program is the National Audiovisual Institute. And that's not by chance. They have a real challenge because they collect not only films and old movies, that's actually how I knew them, but also recent films as well as TV shows from many countries. And they're at several tens of petabytes, 10 to the power of 15 bytes. Today they're at 80 times 10 to the power of 15 bytes, so that's 15 zeros after the 80. It's considerable. It's growing rapidly, so they have a problem. If you want the details, here they are. Their charter requires them to have 3 copies in France, and their budget allows them to have 2. So they found it interesting that DNA allows them to have 1,000 more copies without doing anything. They can distribute them across the territory or use them however they want. So that's one area of application. It's cultural heritage. You can also think of books. You can also think of architecture. You can think of many things. That's one aspect, but we can also think about scientific heritage. CERN, for example, which also has those few dozen petabytes of experimental data that they want to pass on to the physicists of the future. And so they want to preserve them. Or banking archives, since banks are required to keep their clients' data for 10 years, 20 years, 30 years. It depends on which data. So you see that anything related to heritage is interesting as a niche market because precisely it doesn't require too much effort. For writing, rewriting, and constant reading, which works very well with conventional methods. Electronic, computer-based, magnetic, and which for now is done too slowly and too expensively with DNA. There you go.
DNA could actually be a way to store information because we're confident the technologies needed to study and decode it will remain available.
Yes, for any civilization with technology advanced enough in chemistry and biochemistry to read and write DNA, in principle, it's doable. So there have been attempts to send CDs into space, things like that. Well, we haven't gotten any response. We didn't get any answer. But by keeping our feet on the ground, so to speak, we can easily imagine spreading out on many sites the same information about what you want. Francophone literature, as well as, yes, where are the nuclear storage sites, long-term waste sites, or all sorts of things. Yes, it's possible. The advantage of being able to work on many sites comes from the fact that actually when you synthesize DNA, it's not just one DNA molecule that you're synthesizing. It's typically billions or even billions of billions, and it doesn't cost more or just barely more. And under these conditions, you can absolutely just put a few billion here and a few billion there. And actually spread them out by sending a letter with a tiny capsule inside that's about this size to a recipient who will put it somewhere safe or something like that. It becomes easy and cheap to do. There, we have a key advantage, that's for sure.
In the coming years, what kind of progress can we expect in this field?
Multiple. Actually, above all, it's about making the process commercially viable for large-scale applications, like, let's say, some kind of replacement for magnetic tape, the role currently filled by magnetic tape. And so it's really the continued improvements in the speed and cost of reading and writing that will be key. Then there are other aspects. So to answer your question directly, first, there's the possibility of using other polymers besides DNA. DNA, of course, has great longevity and many advantages, but it also has some drawbacks. And from this point of view, other polymers might do a better job than DNA. So we can move away from it to varying degrees. There's an excellent chemist in the United States who actually suggested, in a way, to rectify DNA, so to speak. And so actually, by changing its chemistry a little bit, but still keeping the backbone, basically, he has both a rectified DNA, which doesn't have the minor drawbacks, I'll spare you the details, of natural DNA, firstly, but secondly, it still retains the ability we have to easily amplify it through a process that became very, very famous in 2020 and is called PCR. That is polymerase chain reaction in this case. PCR is a way to read DNA, a way to amplify it. Typically, in a few hours, for just a few cents, you can amplify it 1 billion times and so on.
Just a quick question. Is it by PCR? Is it related to the PCR test?
So,
absolutely.
Let's say that, of course, the PCR test relies exactly on this same polymerase chain reaction, PCR, the English acronym. So they have in common the practice of molecular biology, which actually dates back to the 1980s, but then we use it in different ways. In the test, we use its ability to amplify. And so to amplify the signal as well, and to detect a very small amount of virus in the nose, for example, we amplify it to the point where we can detect it and make it visible. But we can use this amplification to read DNA as well. It can be done. And to get back to the subject, actually, this American chemist has kept the structural characteristics of DNA that allow us to use PCR. We can amplify it by this process. It's simply because DNA is a double helix that is antiparallel and complementary. And so actually we can amplify one of the two strands by making the other strand, by synthesizing the other strand. That's all. That's really what it comes down to. Whereas a chemist from Strasbourg, Jean-François Lutz, proposes polymers that have nothing to do with DNA anymore. And so the only drawback is that we can't do PCR. So we don't have this easy ability to amplify, but the advantages are still interesting. First, it can have different chemistries, sometimes with up to 8 or 16 monomers. And so, even today, with the quaternary DNA alphabet, you encode 2 bits with a single letter, with a single monomer. Then, if you have 16, you encode 4. There you go. So, we're increasing the information density even more. And alongside that, we can have an almost infinite lifespan. Unfortunately, we know this because we have plastic materials in the ocean. In this case, it's not a problem because it's a tiny little fiber that's going to carry the message. We're not talking about lots of plastic bags, of course, but we do have very long lifespans without using special preservation methods like you need for DNA. Because the mammoth's DNA in Siberia was preserved not only because of the cold, but also because it was inside the bone matrix. In the laboratory, the DNA is purified and it is placed in a protective atmosphere, in a very small capsule, for example. And so that's a small difficulty, let's say, that we don't have with certain fibers like that. Afterwards, yes, to also answer your question about the upcoming difficulties, there are the difficulties to read these non-DNA polymers from Professor Lutz in Strasbourg. Today we can read them with machines that are hard to bring into the field because they're still about a cubic meter in size. Well, it's very fast, it's very efficient, but outside the laboratory it's complicated. However, in the long run, we can expect that it will catch up with DNA approaches. which already benefit from devices that can read DNA in the palm of your hand and are connected to your cell phone. With that, you can go anywhere you want, in the fields or elsewhere. And these methods are already very interesting, but they will only keep getting better. And that's the key point that will once again broaden this approach's range of applications, and we hope that happens quickly. I was just talking about a full-scale commercial maturity around 2035, 2038. We're not that far off. We had calculated that basically the conventional method in data centers would hit a very hard wall in 2040. In terms of land footprint as well, these warehouses are eating up land pretty much everywhere in the world. Electricity consumption, as I mentioned earlier, and other drawbacks. And then, of course, since it's also a form of nanotechnology, it's not compostable. So, what do we do with the waste? We have trouble recycling rare earths, silicon, cobalt, and so on, which are in limited supply. So, in light of all this, we estimated that conventional methods will hit a very hard wall around 2040. And DNA and polymer-based solutions, which will arrive in time, if all goes well. There you go.
On a more personal level, what attracted you to this field? What made you want to go into synthetic biology?
All right. Yes, that's an important question, because actually I've asked myself that several times. So what you need to understand is that, above all, I'm a cell biologist. That's been my entire career in basic research. Has been about trying to understand how a cell is organized and how it works, whether we're talking about bacteria or yeast, so microorganisms. And this led me to use approaches that have changed over time. And I used microscopy, I used biochemistry, molecular biology, microbiology, and so on. To get a little closer, around the 1990s, bioinformatics, network biology, bioeconomy, systems biology, and finally, around 2005, synthetic biology. But as a tool to help answer my questions about how living things function and are structured. Also, synthetic biology has a strong appeal. Not only for the basic researcher, but also for the applied researcher who does research and development towards applications that we discussed earlier. And that's why I also reinvented myself. And in addition, as a sort of biotechnologist, I became interested in the possibility of making use of some of our discoveries. Which emerged very clearly from biotechnology or even from synthetic biology. And that's where much of my interest in synthetic biology lies, which I worked on, I think, roughly between 2005 and 2018. Okay, with a lot of educational programs as well, a lot of conferences, thematic schools, and so on, as it should be in the field of public and fundamental research. So there was also something a bit appealing in synthetic biology that's connected to an educational program aimed at young people. Young women who are, let's say, between their final year of high school and their undergraduate degree, so between 17 and 23 years old, roughly. And it's an international competition called iGEM. So international, sure, but GEM actually stands for genetically engineered machines. Genetically, how do you say it in French? Educated or developed? Well, so actually it's a synthetic biology competition that started around 2004 and grew bigger around 2006, 2007. And I was one of the older members in the French team, the first French team, EGEM, in 2007. Afterwards, we went to MIT in the eastern United States, near Boston, for the competition itself in November. And we won a gold medal. We won a lot of things. It was amazing. I have to clarify right away that, in fact, it was mostly the work of the younger members. But the young people were accompanied by a few older folks to, let's say, help them, provide them with lab benches to work at, some advice and so on. So there were a few more mature people, but above all, there were young people who were really excellent. They thought they were excellent. And they really were the reason for the success of the first French team, which led to the creation of other French teams. Then, even now, this year again, there are French teams, several French teams taking part in this kind of competition. You see, and here I think we're getting to a point that really, well, that I personally care about. To create among young people an atmosphere of emulation and an exceptional atmosphere of enthusiasm. This wasn't something that started with synthetic biology. If you go back, for example, to the beginning of the 20th century, it was very much about poetry. And in particular, German speakers were able both to say, I wrote a poem, I did something good, here it is, but also to say, but he's really the best. He's really good at that. It's good to be able, in this context, to recognize the value of others, and that's what was happening. There was also such a spirit, I think, between the two world wars regarding philosophy, gnosis, for example, etc. There was all of that. And it won the hearts of many young people who then went on to do very useful things. In any case, more useful than winning the hearts of young people in order to get them to carry out attacks, it seems to me. Today we are faced with this situation where biology has become a kind of new frontier for some of our young people. On every continent on the planet, the IGM competition is a great driving force for this kind of approach. So that's something that's valuable. And this unique and absolutely exciting experience, but also an absolutely exhausting competition, IJM has made it possible to bring together a lot of people every year for about 20 years around the issues of biotechnology. So that, I think, also helped me see biosynthesis as an important issue. That's it.
So now we're coming to the end of our interview. I also like to talk about the books our guests have written. Here you were able to bring us a few. So two little ones here that are quite fun. Synthetic Biology, Stronger Than Nature. Who is it for?
Question mark.
Question mark.
So unfortunately this book, I don't know if you can see it on camera, already dates back to 2011. Nevertheless, some early successes are noted, even though today we mostly talked about more recent successes in synthetic biology. So it has the advantage of giving a bit of an overview of the topic, including ethical aspects, historical aspects, and so on, in just a few pages. It's a little book published by Le Pommier, as they've done on many subjects. Pour ceux qui préfèrent, you can still find it, but mostly second-hand now.
All right. And so a second book that you brought, Archiving Big Data Beyond 2040, that's also one of the topics we discussed. Do you go into this topic in more depth in this book?
Yes, without a doubt. This book is actually the report from the French Academy of Technologies on the subject. To this day, there is no more comprehensive report in the world. It was also edited and published in English so that more students and more researchers entering the field could make use of it. It also has the drawback of not being completely up to date. It was published in October 2020. So I want to point out that you just have to ask me to get a free copy, and that, moreover, you can download it online in English or in French from the website of the Academy of Technologies. In any case, yes, it is currently the most comprehensive work on this subject. We will consider an update in about a year, because this field changes quickly.
So we'll put the links in the description anyway. We're coming to the end of our interview. Thank you very much, François, for this interview.
Thank you for the invitation.